Can you see my screen?
Yes, please. Yes, please. Just, Professor Arkin, will you give us just a short presentation about yourself? Okay.
Yeah.
So.
Yeah, I prepared one or two sort of very brief words about myself.
Thank you very much.
Sufficient to make my mother happy.
Okay, great. I'm sure they're more than happy about you. But
well,
okay.
I don't know. We'll see. So you're going to tell me when to start, or are we waiting for the first?
No, that's fine. That's fine. You can start. Yeah, that's fine. Pierre will talk after you. Yes, please.
Okay. So thank you very much for inviting me to this exciting e-meeting. I think we're all concerned about the same topic. And before I start, let me just again thank the organizers, say that we are a new small startup, but I've been working in the field of viral proteins for more than 2 decades. But the thing that's probably on everybody's mind is, how is Israel doing? Because that's where I'm located right now, in Jerusalem. So I'm going to show you the next slide, and please, word to the advised, don't get discouraged. This is the vaccination percentages of Israel. Normally we are associated with a public perception of slightly more negative, but this time we are doing phenomenally well. It's mostly, actually the vast majority is the Pfizer messenger RNA vaccine. And I think something like a quarter of the population has been vaccinated twice, and up to 40% have been vaccinated once. So hopefully we will see the end of this scourge in the next few weeks. They've started the vaccination regime from the elderly towards the younger, and since the elderly are less mobile, the number of infections has not dropped, because it's still the young people that are moving about and infecting everyone else. But at least the number of those above 60 years old that have been admitted to the hospital due to COVID has dropped substantially. So that's where we are. So a few words about myself. I got my PhD at Yale University. I was on the faculty of Cambridge University for a few years. And in the year 2000, I moved to the Hebrew University, where I spent, where I've been ever since. I also had a 8-year stint as the vice president of the university, and I also spent a few years in D. Shaw Research, the premier research organization for molecular dynamics simulations. So what are we trying to do? What we are trying to do is to talk about drug repurposing, how it impacts COVID-19, and in particular viral ion channels. So other distinguished panelists will talk about vaccines and other things. I will try to focus on drug therapy, which is one of the 3 approaches to curb a viral disease, the other being vaccination and convalescent therapy. So as I said, the talk will be divided into 3 segments: the idea of drug repurposing, the target that we have chosen, which are viral ion channels, and what have we done with COVID-19. So drug repurposing in general is a relatively simple idea. Take a drug that has been approved by the authorities, normally FDA, and see if it is also efficacious against another indication. Now, why would you want to do that? Because it actually reduces the cost of drug development appreciably. And people have estimated that the cost of bringing a new drug to market is anywhere from $2 to $3 billion. And bringing a new repurposed drug, new indication, old drug, is 1/10 of that, $200,000 to $300,000. The lower cost and faster rate means that this can be applicable also to neglected diseases. So the pharmaceutical industry might be motivated to go after something that is not a huge blockbuster, but rather something that has a smaller patient audience. And most importantly, in the last decade, it's been estimated that a third of all drug approvals have been repurposed drugs, which have contributed to 25% of the revenues in recent years. So this is a very important new avenue of research. And the reason that we have decided to focus on repurposing is simple. Even if today in the laboratory we come up with a brand new chemical that can combat COVID-19, the length of time at which a patient can benefit from that will be many years. And now is the danger is clear and present and momentarily, so we need to look at another route. So here are a few examples of famous repurposed success stories. Minoxidil is one. As you can see, if you look at me, I have not used it. Perhaps I should have. Sildenafil is obviously very famous for erectile dysfunction, also known as Viagra, and thalidomide is actually the #1 drug in the world today for multiple myeloma, despite its horrendous public relations that it suffered due to the pathologies that it impacted. Well, what about viruses? Has drug repurposing been useful in viruses? And the answer is absolutely. AZT or zidovudine was initially described in the 1960s, and it was developed as an anti-cancer. It was a modification of one of the nucleotides, thymidine, but it was actually a total failure. And it took more than 2 decades to realize that this is a wonderful inhibitor of reverse transcriptase in HIV. And now it was able to turn the tide against a dreadful disease. And together with the AZT and other drugs, AIDS has been vanquished in most Western countries. COVID-19, we've seen remdesivir being approved in lightning speed. Obviously, it's still a contentious issue. Is this drug actually useful against COVID-19? But it's important to realize that it was developed more than a decade ago against other viruses, hepatitis C and respiratory syncytial virus. It was not successful. It was transitioned by Gilead, the developers of remdesivir, against Ebola and feline infectious peritonitis. Why is this important? Because feline infectious peritonitis is a coronavirus, which immediately brought the people in Gilead to realize, come COVID-19, perhaps it can be used for that as well. And within less than 6 months, it was approved for use. So this tells you about the speed of drug repurposing. Now, when you think about drug repurposing, you need a particular target. What will you be targeting in the virus? And our focus has been on viral ion channels. Why ion channels? Well, first of all, they're very common components in viruses that don't receive a lot of attention. They're present, you can see the list of viruses over here, both in enveloped and non-enveloped viruses, both in RNA and DNA viruses. And again, the most famous one, the archetypical viral ion channel, comes from influenza, and that's actually the target of aminoadamantine, simitril and flumidine. The biological function of viral ion channels is to enable the pathogen to regulate its salinity and acidity of its environment and its surroundings and its internal environment. And because of that, it might serve as a very attractive drug targets. Now, ion channels as a family are fantastic drug targets. Nearly every drug that targets hypertension, anxiety, most anesthetics, etc., target ion channels in our body. But ion channels in viruses have been underexploited, so that's what we want to target. So this is what ViralBlock, a small company that we have established based on the decades of work that I've been doing on this, to try to provide rapid solution to viral diseases. And obviously today, our focus is on COVID-19. So what we have developed are assays in which one can test the activity of these channels and to identify inhibitors thereof. And the reasons that these assays are attractive is because we study them inside a biological membrane of a bacteria. Now, this enables us to rapidly change our system, to evaluate different mutations, and to test thousands of compounds within a month or so. So this is a very, very simple and robust system. And using the power of bacterial genetics, we can also talk about drug resistance potential, but I won't speak about that today. So what is the task at hand? The first thing that we need to do is to ask ourselves, does this new virus have an ion channel? And sometimes it already has one, but we need to confirm that it actually works in our particular assays. The second thing that we need to do is we need to screen for blockers. Once we've developed an assay, we can screen for blockers. And as I said, we screen drug repurposed compounds, a library of about 3,000 chemicals that one can purchase commercially. And finally, you can assess resistance potential. And once you identify the resistance potential, you can have in a reciprocal manner to actually test new compounds, etc. So one or two words about how we actually test these. And again, if any of the audience is not a bacteriologist, please forgive me if I'm speaking, if I sound too technical. But the idea is relatively simple. Here you have nice, thriving bacteria on a petri dish. You express the viral channel inside the petri dish. And because it's a channel, it permeabilizes the bacterial membrane. It makes lots of holes in the bacterial membrane. And because of that, the bacteria are dead. All you need to do is to grow lots of these bacteria in individual wells in a robotic system, which we have. And each of those wells you treat with a different chemical. And if some of these bacteria survive, it's because you were able to block the viral ion channel that is actually permeabilizing the bacterial membrane. And on the right, you can see exactly a result of such an experiment. This is the growth rate of happy bacteria that do not express the viral channel in red. In blue are the growth rate or lack thereof of bacteria that express the channel. And here is the growth rate of bacteria in which you were able to identify a blocker that inhibits the channel. So that's a good sign. We also have another assay that is entirely reciprocal to test the validity of our results. So here we identify the channels using an entirely opposite approach. Here we have a bacteria that are deficient in a particular channel. And when you grow these bacteria in the presence of low potassium, they are unable to grow unless they express the channel. So in this instance, the viral channel is beneficial to the bacteria, and the antiviral drug will have the opposite approach, the opposite impact. So you can see exactly here, you can see here bacteria that are not expressing viral ion channel are non-viable. When they express the channel here in blue, they are viable, but when you block the channel, once again, they're unable to grow. We have a third assay, I don't want to go into that, but it's based on fluorescence, etc. So what have we done? The first thing that we've done is we've shown that there are two channels, two targets in SARS-CoV-2, the E protein and the open reading frame 3A. And in all honesty, this was not a surprise because we are familiar with both of these proteins from the days of SARS. SARS that was identified in 2003, but received very little attention because the pandemic that it responsible for basically vanished. We have screened 3,000 chemicals, each of which has been approved for use in humans, but for another indication. And then we have identified about 10 inhibitors of each of these channels, and we were able to assay the antiviral activity against the whole virus in a biosafety level 3 lab. So this is what some of the results look like. Again, I won't bore you with the technical details. These are the different hits. And here you can see increased concentration of the drug enables the bacteria to thrive, to grow faster, because these bacteria are currently suffering due to the viral protein. In the opposite assay, the opposite thing happens. increased concentrations of the drug causes the bacteria to grow slower, because in this assay, the viral protein is beneficial. So we are convinced that if you get a positive outcome in both of these assays, these hits are probably real. And this is the final outcome. This is the result. So here we tested in tissue cultures the ability of viruses to replicate. After 48 hours, these compounds were added at a relatively high concentration, admittedly. And as you can see, most of them are able to inhibit viral replication by anywhere from 40 to close to 100%. So we are very excited about these results. And again, each and every one of these compounds have already been approved for use by the FDA against another indication. And what we are trying to do right now is to trying to start clinical trials. We can forego clinical trials at phase 1 because the toxicity of all of these compounds has already been established. So either 1B or 2, and that's precisely where we are right now. So again, the next step, we're trying to forge an alliance with pharmaceutical companies because they are best suited to develop these findings further with clinical trials. We're also interested in engaging NGOs about diseases other than COVID-19, such as dengue, Zika, etc. And we are also interested in attracting pharmaceutical companies to give us their compound libraries that we might be able to screen. So with that, I will finish, and I hope that was sufficiently understandable to the non-professional audience, and I'll be very happy to take any questions if that is appropriate at this time. Oh, I should stop sharing. Should I?
I think I hear you, Ferry. Sorry, I had a problem with the microphone. It was mute and I couldn't change it. Thank you very much. Thank you very much. So do you think that all of the doctors, they have the possibility to prescribe these medications, these treatments easily for the patients, or these are only used at hospital level? Because we do not really hear that the doctors have a lot of possibilities nowadays, at least here in France, to prescribe any treatment.
So in terms of antivirals per se, so compounds that actually inhibit the viral replication rather than subdue your immune response, you're right. I think the only option today is remdesivir. And I think because it needs to be taken intravenously and the cost is substantial and the benefit is minimal at best, it's really not an option. But I think things like dexamethasone, which allows our immune system to be underchecked and not go into a cytokine storm, has proven to be very, very effective. But again, I wouldn't consider dexamethasone or any other such compounds as an antiviral agent. But again, what we're trying to do is to push these through further through clinical trials, and hopefully we'll have some news in the next few weeks. But again, these are, well, by that I mean that we are trying to engage people to fund these research studies because they're quite costly.
Mm-hmm. Okay, thank you very much. So still we do not have really a lot of options for treatments, not yet, but in the upcoming weeks we will have more options. That's what I understood.
I hope so. And there's other, there are other repurposing studies against COVID-19. There's a group, a very, very strong and prominent group in Scrip Institutes in San Diego that are doing amazing work. They're not targeting the ion channels, they're targeting other elements in the virus. So I'm hopeful that things will simmer through and we will have compounds to add to our arsenal of anti-COVID medications.
Okay, we really need it. That will be a great hope, you know, to the humanity, to humanity.
Yeah, I agree.
Thank you very much, Professor Arkin, we are really honored to accepting our invitation. Will you please now share your role with Pierre Morgan? I think that as Professor Stegman is having trouble with his laptop, he sent me a message. So we will proceed by Pierre Morgan. So you have to choose him as the host, new host. So
I made you the host. You are now the host, I believe.
Okay. So let me share it. No, I cannot do it anymore. Okay. Nothing. Pierre, can you share your screen? I gave it, you know, I gave you and your microphone is mute, Pierre.
Yes, because now it is unmuted. You know, I was trying desperately to activate